Hello friends, welcome to the channel of Pharma. uh, Myself Vijay. Today we see the ICS guidelines. Uh, ICS guideline consists of the quality, safety, efficacy, and multidisciplinary. We see the firstly quality, different types of guidelines in quality. uh, First one is the ICS Q1A. ICS Q1 on a stability. We ICS Q1A R2 stability testing of new drug substance and new drug products. This guideline applicable for the new drug substance and drugs products. Then ICS Q1B stability testing. In stability testing consists of the photo stability testing of new drug substances and products. Then ICS Q1C stability testing of for new drug form. Then ICS Q1D bracketing and matrixing designs for stability testing of new drug products substances. Then ICS Q1E evaluation of stability data. ICH Key1 stability data package for registration application in climatic zones 3 and 4. Then ICS EWG stability testing of the druster and drug product. These are guidelines for the ICH Q1A. Then ICH Q2. This consists of the analytical validation Q2 R2 revision of R Q2 R1 analytical validation. This guidelines applicable for the analytical method validation for new drug substances. Then Q2 R2 Q14 training on validation of analytical process. Q14 analytical process development. These are new guidelines for analytical process development. This is the Q14. Then ICH Q3A Q3E impurities. These are guidelines for the impurities in for pharmaceutical product. This consists of the Q3A1 Q3R2 impurities in new drug substances. Then Q3BR2 impurities in new drugs products. These drugs products consist of the impurities also calculated based on the uh, manufacturing process. Then excipients. These are cumulative impurities are specified in specification of the drug product specification, and these are included in specification and tested during the routine release of the batches. Then ICS Q3C, that's the guideline for the residual solvent. Then ICS Q3C R10 maintenance CWG maintenance for the guidance of residual solvent. Then ICS Q3D guideline for elemental impurities. The ICS Q3D R3 maintenance of guideline for the elemental impurities. Then Q33 training implementation guideline of elemental impurities. Then Q3 EWG guideline for the extractable and leachables. In impurities mainly consist of the residual solvent, elemental impurities, then the extractable and leachables. Then ICS Q4A and Q4B pharmacopoeia Q4A pharmacopoeial harmonization. Then Q4B evaluation and recommendation of pharmacopoeial test by using ICS region. Then Q4B R1 evaluation of recommendation of pharmacopoeial test for the using ICS regions. Q4B Annexer 1 residue on ignition sulfatase as general chapter. Q4B Annexer test for extractable volume of parental preparation central chapter. Q4B Annex test for the particle contamination is a visual particle general chapter. Q4B Annex 4 R1 microbial examination of non-sterile products microbial enumeration test general chapter. Q4B Annex Q4B R1 microbial examination of non-sterile products test for specified microorganisms general chapter. Q4B Annex Q4C R1 microbial examination of non-sterile products acceptance criteria for pharmaceutical preparation substances for pharmaceutical use general chapter. Q4B Annexer 5 R1 disintegration test general chapter. Then Q4B Annexer C uniformity of doses units general chapter. Q4B Annex 7 R2 resolution test general chapter. Q4B Annexer 81 stability test general chapter. Q4B Annexer 9 R1 tablet priority general chapter. Q4B Annexer 10 polyacrylamide gel electrophoresis general chapter. Q4B Annex Capillary Electrophoresis General Chapter. Q4B Annex 12 Analytical Sieving General Chapter. Q4 Annex 13 Body Density Tap Density Powder General Chapter. Q4B Annex Bacterial Introduction Test General Chapter. Then Q4B Frequently Asked Question. Then Q5A Q5 Quality of Biotechnology Product. Q5 R2 consists of the viral safety evaluation of biotechnology products derived from cell lines of human and animal origin. Q5A R2 testing training on viral safety evaluation of biotechnology products derived from cell lines of human and animal origin. Q5B analysis of expression construction in cells used for production of rDNA derived protein products. Then Q53 quality of biotechnology products, stability testing of biotechnology and biological products. Q53 derivation and characterization of substance used in production of biotechnology and biological products. Q5A compatibility of biotechnological products subject to changes in their manufacturing process. Then Q6A Q6B specification. This Q6 guideline applicable specifications of drug substance and it mainly consists of the Q6A, this is specification, test procedure and acceptance criteria for new drug substances, new drug products, chemical substances. Q3, Q6B specification, test procedure and acceptance criteria for biotechnological and biological products. Then Q6R1 AWG revision of specification guidelines. Q7 good manufacturing practice. Q7 is the applicable good manufacturing practice. Q7 good manufacturing practice guide for active substances. These are guidelines applicable to the manufacturing plot of API. Then Q7 question and answer. अभी Q8 pharmaceutical, this is a guideline for the pharmaceutical development for new disease form. This mainly consists of Q8 R2 pharmaceutical development. Then Q9 is a quality risk management. This consists of Q9 R1 quality risk management, Q9 R1 IWG, the training on quality risk management, the Q9 R1 training policy quality risk management, and then Q2 other फार्मास्यूटिकल क्वालिटी मैनेजमेंट अभी इसमें क्वालिटी में जो भी है मैन्युफैक्चरिंग में हम चेंज कंट्रोल डेविएशन इंसिडेंट्स इंक्लूड होते सभी गाइडलाइन के रेफरेंस हम वो सब फॉलो करते हैं. इसमें आता है क्यूटी में फार्मास्यूटिकल क्वालिटी सिस्टम. देन क्यूएलएन डेवलपमेंट एंड मैन्युफैक्चर ऑफ ड्रग सब्सटेंस. अभी क्यूएलएन गाइडलाइन रेफरेंस गाइडलाइन डेवलपमेंट ऑफ एंड मैन्युफैक्चर ऑफ ड्रग सब्सटेंस के लिए. क्यूएलएन में आता है डेवलपमेंट मैन्युफैक्चरिंग ड्रग सब्सटेंस केमिकल इंटेंस बायोटेक्नोलॉजी एंड बायोलॉजिकल एंटिटीज. Then Q2 आता है rep cycle management. Q2 technical and regulatory consideration pharmaceutical product life cycle management. Then इसका ट्रेनिंग आता है Q12. अभी जो भी कंटिन्यूस मैन्युफैक्चरिंग ऑफ ड्रग सब्सटेंस और ड्रग प्रोडक्ट है, अभी जब भी हम ड्रग प्रोडक्ट बनाते हैं बैच वाइज बनाते हैं, पर जब भी हम कंटिन्यूस मैन्युफैक्चरिंग अप्लीबल करते हैं उसके लिए गाइडलाइन है Q3 Q13 कंटिन्यूस मैन्युफैक्चरिंग ऑफ ड्रग सब्सटेंस एंड ड्रग प्रोडक्ट्स. देन Q14 आता है, अभी ये नया गाइडलाइन है एनालिटिकल मेथड वैलिडेशन के लिए, एनालिटिकल प्रोसेस डेवलपमेंट. Q14 एनालिटिकल प्रोसेस डेवलपमेंट एंड Q2 R2 Q14 ये दोनों गाइडलाइन अवेलेबल है एनालिटिकल प्रोसेस डेवलपमेंट फॉर द न्यू ड्रग सब्सटेंसेस. थैंक यू.